Welcome to Botany class. Let's continue microscopy. In the previous class, you learned two different types of microscope. In your syllabus, there are four different types of microscopes. They are bright field microscope, bright field microscope. The first type of microscope you learned is bright field microscope. Then dark field microscope. These two types already you have learned. Now third type of microscope is called as phase contrast microscope. Phase contrast microscope and electron microscope. Electron microscope. There are four different types of microscopes. What are they? Bright field microscope, dark field microscope, phase contrast microscope and electron microscope. The electron microscope, again it is classified into transmission electron microscope, and scanning electron microscope. There are two different types of electron microscope. Electron microscope has two types: transmission electron microscope and scanning electron microscope. Let's start from phase contrast microscope. I hope that you learned first two types of microscope, bright field microscope as well as dark field microscope. Now phase contrast microscope. The phase contrast microscope, see this is the light path in phase contrast microscope. It was invented by Cernic. The phase contrast microscope was invented by Cernic. Cernic invented phase contrast microscope. This is the modification of light microscope with all its basic principle. Basically, the principle is resembled to light microscope, that is bright field microscope, but there is a modification. The object is viewed by amplified the light waves. The light waves are amplified and the contrast is increased. The contrast from the specimen is increased in phase contrast microscope. How it is increased in phase contrast microscope? Here there is introduced by a phase plate. The name of the microscope is phase contrast microscope. Here the contrast is increased by introducing a phase plate. So look at this diagram. This is the light path in phase contrast microscope. Here this is the region that introduce the phase plate. The phase plate is a circular annular etching. So etching is a process of printing process. They are using this technique and printing this image by means of this phase plate. So phase plate increases the contrast. So this is used in this. When this light is, the phase plate is increased in the condenser. So when the light is passed through, the beam of light, when it passes through this specimen, so you know in the microscope there is a specimen. In light microscope we used to place it in the slide, microscopic slide. The specimen is spotted there and mounted it cover slip, and it is placed on the stage of this adjustable stage present in the microscope. Here this is the stage. So light is passing through amplified, which is amplified light waves. When it passes through the condenser, here there is a condenser. So objective is present here. So there is a cone of light pass through from the specimen. So when it is passing through face plate, here already it is introducing a face plate to increase the contrast. When these beams of light pass through this face plate, there are two different regions, thick region as well as thin region. In the thick region and the thin region, the direct light is passed through the thin area, and the specimen, from the specimen, the light is passing through the thick area. So when this thin area light is passing, the velocity of light is low velocity, low speed through the thin, that is direct light, it goes as a low speed, and from the thick area of this face plate, when the light is passed through, it goes as a low speed. So there high speed, here it moves as a low speed, and this detects the image in this image plane. Here there are cones of light, there is a cone of light which visualize the image in the image plane. This is the principle it works, and this phase contrast microscope is used to observe the cells and the tissues, and in vitro culture at the stage of mitosis, the tissues when they are cultured in the in vitro during mitosis stage. So these all cells and tissues are visualized by this phase contrast microscope. So this is the first type of microscope you are going to learn today. Second type of microscope is electron microscope. Now in electron microscope, instead of this light beams, here the light beams are used. The light waves are used to contrast that increase it shows the image. Light beams are used. Instead of light beams, in electron microscope, here there is electron. The source of illumination in electron microscope is electrons. In electron microscope, the source of illumination by means of electrons. So beam of electron, like this light instead of this light, beams of electrons pass through the specimen. The object is viewed by means of passing directly through the specimen. The electron beams are passed through. So this electron, there are opaque chemicals are used to dehydrate the specimen. When the specimens are used in electron microscope, there are two different types of electron microscope, transmission electron microscope as well as scanning electron microscope. In transmission electron microscope, TEM, transmission electron microscope, it shows a two-dimensional image. So when the specimens are mounted, here the specimen is dehydrated specimen with opaque chemicals like gold or palladium. The gold or palladium is used as a opaque chemicals to dehydrating the substance, that is the object, the specimen. And the image is 1 lakh to 3 lakhs, 1 lakh times. 1 lakh times the image is contrast. So resolving power is more, high velocity. So here the transmission electron microscope, it resolving power is, the magnification is 1 lakh time more than that of the light microscope. And in this transmission electron microscope, there are some components. The components are electron generating system, which consists of electron generating system. A electron generating system is used in this transmission electron microscope. By the name itself, you understand electron microscope, there is the electron generating system is operated, and electron condenser, instead of normal condenser, electron condenser is used, and the specimen objective is used, and tube lens and a projector. A tube lens, projector. What are the components? These are the components which is present in transmission electron microscope. Electron generating system. The first component is called as electron generating system, and electron condenser. Instead of normal condenser, here electron condensers are used. And the specimen objective, tube lens as well as the projector. So, the image is screen on the fluorescent screen. The image is seen on the fluorescent screen. And transmission electron microscope used to view the structures of virus, mycoplasma and so on. Also the cell organelles. Very fine image. Here it is electron microscope, we can say this ultra microscope, ultra structures. Ultra structure is visible. Very minute structures, cell organelles, viruses. So, this type of all these minute organisms or organelles can be visualized by means of electron microscope. One among this electron microscope is transmission electron microscope. Another microscope is scanning electron microscope. In scanning electron microscope, it is called as SEM, scanning electron microscope. This is transmission electron microscope. In scanning electron microscope, the image is 2 lakhs time more than that of transmission electron microscope. In transmission electron microscope, 1 to 3 lakhs time, 1 lakh time it is magnified. But here, scanning electron microscope, there are 2 lakhs times, and the resolving power is lower than that of transmission electron microscope. The resolving power in transmission electron microscope is more, and the resolving power of scanning electron microscope is less. That is the resolving power 2 to 10 Armstrong unit in transmission electron microscope, and 5 to 20 Armstrong nanometer, 5 to 20 nanometer in the scanning electron microscope. In the scanning electron microscope, the electrons are captured or focused by means of lenses. In this microscope, scanning electron microscope, the electrons are focused by means of lenses. So, lenses which focus the electrons, and it views in a point. So, there is a interaction between the specimen as well as the electron. When the interaction of specimen and the electrons, from the specimen there are some radiations released. The specimen which, when these electrons are interact with the specimen which releases some kinds of radiation in the form of auger electron, secondary electron, or back scattered electron. These are the different types of electrons which release from the specimen. Now, these electrons are captured by a detector. It's captured by a detector, and this detector, from the detector, the image is viewed in the fluorescent screen. On the fluorescent screen, the image can be visualized. This is the principle the scanning electron microscope works. So, 3 different types of microscope you will learn today. One is phase contrast microscope, another one electron microscope. 2 main types. Under electron microscope, there is a transmission electron microscope as well as scanning electron microscope. Now, what is the comparison? Now, you are going to compare comparison of these all kinds of microscope. You have learned 4 different types of microscope, that is bright field microscope, dark field microscope, electron microscope, transmission electron as well as scanning electron microscope. So, what are the features of this microscope? There are several features. First feature is the illumination, the source of illumination. The source of illumination among these 4 microscopes came from only 2 sources, that is visible light or the electron. So, first 3 microscopes, the first 2, this bright field microscope and dark field microscope, the light is visible light. The source of illumination is visible. But in electron microscope, both transmission as well as scanning electron microscope, it is electron. And the electron microscope is introduced by Ernest Ruska and which is developed by Binning and Roher. Binning and Roher developed this microscope, electron microscope, which is introduced by Ernest Ruska. The second feature of this microscope is types of cell visualized. How the types of cells are visualized. In ordinary microscope, that is light microscope or the dark field. Light microscope is otherwise called bright field microscope. First 2 types, the individual cells are visualized. But in electron microscope, thin sections of the specimens can be visualized. The very thin sections of the specimen can be visualized in electron microscope. Then image, all this up to this transmission electron microscope, the image is 2-dimensional. But only scanning electron microscope, the image is 3-dimensional. And the nature of lenses, all these lenses are glass lenses except in electron microscope. Electron microscope, the electrostatic lenses can be used. Electrostatic lenses can be used in electron microscope. Ordinary glass lenses are used in other microscopes. Then the medium. Medium taken for this is normal microscope, air or oil, but in this electron microscope, it is vacuum. The medium is vacuum. And specimen mounting, how the specimen is mounted. Mounting of specimen, there is a glass slide. You know that ordinary microscope, there is a glass slide, microscopic slide, which contains a cover slip. In the center you may find there is a spot, that is a specimen. Through your naked eye we cannot see that, only a spot is present. But when you keep it under microscope and focus it, you may visualize the image of this particular specimen. So, specimen mounting. Glass slides are used for bright field microscope as well as dark field microscope. And for this specimen, the electron microscope, it is used copper grids. In transmission electron microscope, specimens are mounted in a copper grid. And scanning electron microscope, there is a gold coated specimen. The specimen is coated with gold. Then focusing and magnification. The focusing, it changed with the objectives. We can change when you are focusing the image, focusing magnification, the lenses can be adjusted. You may practice it in the lab. If the specimen is not visualized, we are changing the objects here and there, we can move and adjust this. In electron microscope, there is electrical deflection coil is used for focusing and magnification. Electrical deflection coil is used. Then specimen contrast. The specimen contrast is light diffraction. In the ordinary microscope, light diffraction is the contrast. The contrast image from the specimen, the contrast can be increased. So, a very fine structure can be visualized. And in electron microscope, electron scattering technique is used. Electron scattering. The electrons are scattered and the view is visualized. Specimen contrast. These are the specimen contrast. So, once again, comparison. First you learn all the features. When you learn this question, comparison of microscope. So today's topic is comparison of microscope. First you learn all the 4 types of microscope. Bright field microscope, each type they may ask for the exam. Each type is 5 mark question. Bright field microscope, dark field microscope, phase contrast microscope, electron microscope. Electron microscope, there are 2 different types, transmission electron microscope as well as scanning electron microscope. And this microscope, phase contrast, the diagram is given in the book, very neat diagram that shows the light path in phase contrast microscope. Draw this diagram in the class note and learn this question, phase contrast microscope, then scanning, then electron microscope. Types of electron microscope, there are 2 types, transmission electron microscope as well as scanning electron microscope. Finally, there is a question, comparison of microscope. There is a tablet column given in your book. First it shows the features of the microscope and all the different types are given. Bright field microscope, then dark field microscope, phase contrast microscope, electron microscope, under this transmission electron microscope as well as scanning electron microscope. Learn all these questions well. Thank you.